Today on The Future of Everything, the future of statistics. Now, statistics is a field that studies how to collect, analyze, interpret, and present data. There are many definitions, of course. But in this era of big data, statistics and statisticians are on our minds more often, perhaps, than they used to be. There are a lot of data available now, and there's a need to understand and turn data into information, knowledge, and insight. To take an example that's, I think, contemporary, in the setting of the current COVID pandemic, we are watching data about testing, hospitalization, and deaths, and there are many disagreements about how to interpret these properly and inform policymakers who need to understand these statistics in order to make good decisions. One of the challenges of statistics is that it's not always intuitive to the untrained, and sometimes surprising conclusions based on statistical analysis may be difficult to understand. I hesitated to use this quote, but it's just a great quote that I'm going to remind us that a very famous, but perhaps also unfair quote about statistics that I think is attributed to Benjamin Disraeli, who said there are three types of lies: lies, damn lies, and statistics. Now, that's a throwaway one-liner, but it does capture the frustration that some people have in understanding the variations and uncertainties that sometimes, in fact, almost always arise in statistical analysis. Well, Susan Holmes is a professor of statistics at Stanford University, and she's a statistician who has developed general methods for analysis with a focus on biology and medical applications, as well as other areas, and a passion for increasing the understanding, accessibility, and appreciation of statistics among scientists, doctors, and others. So Susan, just to start out, what attracted you to statistics, and what do you see as some of the most exciting current challenges?
I wanted to do mathematics, but as it applies to the real world. And actually, I left a mathematics department because I was asking them to have a computer. And they told me that there would never be a computer in the math building. So they said, if you want to do mathematics and use a computer, you have to go and do statistics. And I didn't even know what statistics was, but I learned statistics. And I never looked back. I'm very happy. I wanted to make an impact. So I think being a numbers person, of course, today, all the more we need, people understand numbers and can explain the numbers to the policymakers. Unfortunately, in the current, situation with COVID, we realize that there's a big gap between the refinement with which statisticians understand the numbers and their third or fourth order approximations, and actually what's being understood by the policymakers. So I try to bridge that gap. That's quite challenging, actually.
I know that you're actually very passionate about you've written about putting tools in the hands of those who have the data, not making the statistician kind of a priest who comes down and blesses the data. So how does that operationalize? What does that mean for your research or for the activities that you partake in when you're trying to kind of democratize access to these kind of pretty high-powered tools?
Well, I would say that it means that I work sort of embedded in a team. That is, I work with the biologists and I actually go in and see how they collect the data. And then I try to understand what they want to do with the data. And usually I do a first pass of the analysis myself with the postdocs, and we analyze the data as a statistician would. And then we present it and work with the biologists to try and see how they could do that themselves. And so that means teaching, of course, biologists how to program and how to use tools like open source tools like R that I use a lot. And then giving them the satisfaction. I think the first thing is showing them how to visualize and plot their data, make aesthetic plots, nice plots, which involve complicated networks and trees and real structures that they have an understanding for, and show them that it's not that bad. And the new generation of biologists are certainly much more open to that. And they see the power of statistics. I think that the old school statistics was seen as just, you do ANOVA or t-tests or something. And that's no longer the case. But then the new generation of biologists come in, we see the undergraduates at Stanford, they've all taken CS 106, and they know
which
how
is
to
the
program.
introduction to computer science.
Introduction to computer science. So they know how to analyze, they know how to write a program. So we have a lot of biologists, especially right now with the lockdown. We had to do 2 special boot camps for wet lab scientists caught just at home with a computer.
So it sounds like you're one of your goals, and it sounds like you've achieved this, is having the analysis be anything but a black box. That if you're engaging the people who collected the data and whose livelihood almost literally depends on a good analysis, then that's a whole different thing. They must have I'm guessing that they like this a lot, and that engages them. Does it ever change the analysis? So you described how you do your kind of professional statistician analysis and then bring it to your collaborators. Does that process of teaching them, does it just get them up to speed on what you already did, or does it sometimes evolve the analysis in ways that you might not have expected?
Oh yeah, because what they bring, and the reason for making the biologists do it, is they have the questions, they understand. And many times the first things that we find, like groupings of cells or something, they just say, oh yes, that's so-and-so. They have an explanation of the cell type or an underlying biological explanation that we saw in the data, but they already knew about. And that doesn't matter. I mean, that just confirms that the data are good. And then
it's kind of a positive validation, isn't it?
Right. But that's not a problem. But then they get hooked on, oh, we could look at this angle or this, as I was saying, this network, and they get involved in the analysis. And they bring also, we connect the data to databases. So they say, oh, well, if you could use this interaction, which is known, and could we sort of map that onto what we're seeing. So it's combining information. But the human part, I think, is really important. We're not yet, I work in an area where we don't have very many samples or many subjects. I work a lot in immunology and the microbiome. We never have enough data to do sort of full-scale machine learning, or when you have 10 subjects or 10, that's not possible. You can really only, you have to explore and take out as much noise as possible, but we can't really use the data in an automated way.
This is The Future of Everything. I'm Russ Altman. I'm speaking with Susan Holmes about the joys of statistical analysis. We're going to get to some of these applications that you referred to in a minute. But before that, you talked about visualization and how the scientists get very excited about looking at their data in different ways. And I know that one of the things your group does is not just do kind of a one-off analysis for an individual scientist, but based on these interactions, you create general purpose software tools that you're very generous in sharing with the community. So how do you move from like scientist X liked this to maybe we have a general purpose tool that we could put out into the public for lots of other uses, even some that we might not even anticipate?
Well, I use the infrastructure which already exists in what's called R and Bioconductor, which is very open. And we have the code. And so we write on top of infrastructure, and we have complicated, today we look at many layers of information in the same way that, the geographic information systems look at many layers of a landscape. So you could look at the roads, but you could also look at the topography and the soil. And so you have many maps. And it's the same thing in what I do. We call it multiomics, or we call it, multi-table, multi-way array. You have many layers of information. And we want to make it so that people can map that information in an easy way and switch back and forth between the maps and the data. And it's quite complicated, but we try to make it so it's just a generalization of things that they've already seen.
Yes. And so once you reach that level, then other people can take different data sets, but apply the same principles and use these tools for their own purposes.
Right. Absolutely. And so the basic, networks are very popular. So you can use networks for gene interactions, but you can also, we use networks for microbial communities or networks for metabolic interactions. So networks is a sort of common denominator.
Yes.
And so if you have that infrastructure, which allows you to say, this network is just like this other network, you have a similarity, that's a tool which carries throughout modern biology. It doesn't matter what the network was.
Great. So let's dive right into that area, because I know this is one area that you've been doing a lot of work, and this is bacterial biology, and specifically, I think, as you said, these communities, or they call them microbial communities. Some people call them the microbiome. What attracted you to that, and what are as a statistician, why does it fascinate you?
Well, I guess I started off in biology. One of my fascinations was actually evolutionary trees and phylogenies. And a research group at Stanford, led by David Relman, was looking, at the time we were doing Sanger sequencing.
This is of DNA sequence technique.
It's an old-fashioned one. This was from 15 years ago, and we wanted to try to make a history or phylogenetic tree of different bacteria and see how that the trees changed across time for different patients. So we wanted to do a comparison. That's how it started off.
So this is based on the idea that an individual patient with some bacteria, those bacteria don't stay the same. They evolve, they continue to evolve even while they're dwelling within the patient. And that might tell you something about the biology of the patient and the biology of the bacteria. Is that,
right. Yeah, that's true. But also what people maybe don't realize is that we have more bacterial cells in our body than we have human cells. So it's a huge number of cells involved, and the large number of communities called the microbiome have an influence on a lot of aspects of our health. And so I thought, as a statistician, this is a wonderful multivariate, multi-dimensional, multi-table, complex problem, and nobody understands the beginning of how to tackle it. So I thought it was a great challenge for having different windows in the same data. Yes. And also it had clinical impact. So we work on many problems that have clinical impact, which have to do with the microbiome. So since then, so for more than 15 years, I've been working with Relman's group on many, many different aspects of what we call microbiome. Now, of course, the thing is, technologies move very fast, and they move much faster than the statisticians can keep up. It takes us about 2 years to figure out how to denoise carefully and normalize the data, and the biologists have already changed their technology. Happily, some of the principles are actually the same, so we can pick up and move on very quickly. But that's a sort of contrary, because statistics in some sense is quite a staid subject. It stays very static and very slow moving, I would say, and then biology is the opposite. So you have to move.
So going back to your collaborations with Dr. Relman, what are some of the things? I know that people are very interested in the microbiome. It's even in the popular press. Can you tell us about some of the interesting findings that have emerged from those collaborations? You mentioned there was some clinical relevance. I'm sure people would be very interested in that.
Well, I would say that one project that we work on, which was very interesting, has to do with preterm birth and the diagnosis of preterm birth. And so we can collect samples of women that are pregnant, just say, collect samples every week of what's called the vaginal microbiome. And these bacteria actually have a signature. And we were able to see that certain signatures were associated to preterm birth. Now, I say, as a statistician, there's a buzzword there. It's associated to, because we don't know, actually, the causality has not been proven. Right. That is, we don't know whether the bacteria are causing the preterm birth, or whether they're just associated to a condition of the immune system or of the environment, which is causing the preterm birth. But we definitely have methods for diagnosis. of high-risk pregnancies, which are now, in translational situation that we can already use. So we have these biomarkers, and these biomarkers are bacteria, and they're very easy to find. And
so you're just so I understand, you're saying that even before the preterm birth happens, you can measure ahead of time changes in the bacterial makeup in the vaginal microbiome, and that's almost like a predictor. What's not clear is whether it's the cause of the preterm birth or some kind of canary in the coal mine type situation.
Right. Exactly.
So that's fascinating. What would be, did the biologists understand why this would happen?
It's an interesting phenomena because in general for the microbiome, diversity is a sign of good health. So for instance, in the gut, if you have a very diverse microbiome with many communities and high diversity, that's usually a very healthy sign. In the vaginal microbiome, it's the inverse. That is, there are special types of bacteria called lactobacillus crispatus and iners. They're protective in the sense of, if they are there, that's a good sign for the pregnancy. That's how it should be. And it shows that there's, the right chemical combination which is occurring, which seems to be protective of the pregnancy. And if there is very high diversity, it shows that you don't have this dominant lactobacillus. And in that very high diversity, then there are certain bacteria which are especially bad or pathological, associated to high risks of a preterm birth. And so Gardnerella are some of them. And with the Gates Foundation, we're trying to find out whether we could actually, whether you can make an intervention. That is, whether you could rectify. And so that you need a whole chemical environment. So it's not only the bacteria, but you also have to change, the chemicals which are present.
Yes, I mean, we've heard about these things called fecal transplants where they replace people's poop, basically.
Right.
Forgive my jargon, but in order to create a more stable colonic microbiome, and it might be that a similar idea might be used here. Is that generally the idea?
Yes, yes. But it might be that we don't even have to use samples from people because it's so simple. The lactobacilli, we have a handle on the strains, and some of them we can actually cultivate. So it's possible to bring this change and, bring about these changes. And we use, there are other ideas about probiotics and how you intervene, but it's definitely, there are clinical outcomes that will come from this, and we have a much better understanding, and so that's one of the projects which has clinical.
No, incredibly exciting, and here, even in this conversation, we've moved from lofty statistical ideas to extremely practical interventions that might be life-altering for both moms and babies.
Right.
So this is The Future of Everything. I'm Russ Altman. More with Professor Susan Holmes about her work in statistics and its interface with biological and other problems next on Sirius XM. Welcome back to The Future of Everything. I'm Russ Altman. I'm speaking with Professor Susan Holmes. We were talking about her work in microbiome, the study of microbial communities, and her work as a statistician. So fascinating work on the microbiome. And I wonder, though, stepping back, as a statistician, what were the interesting challenges or interesting methodological approaches that you brought to this microbiome work?
Well, it was a pretty difficult problem because of the reads were difficult to denoise. So first of all, if you can think about what happens is we created a method which is very much like how Google corrects your spelling. When you do a search and you misspell a word, but a word which is very frequent, but you misspell it, it will suggest another word. So we've created a method for denoising the data, which uses that actually, when we wrote it, we didn't realize that it was the same as what Google was doing, but we used the frequency of the reads. And
when you say read, I just want it for people who are not, these are the small snippets of DNA sequence that come out of a sequencing machine. And of course, you're looking at these bacterial communities and you're sequencing their genomes in order to figure out who's there and who's not there. So these reads are the DNA sequences, which are letters, the 4 A, G, C, T DNA letters.
Right.
Okay.
And the reads that we work with, they're only about 250 characters long.
Okay.
So those are sort of our words. And we created a denoising method called Data2 with Ben Callahan, who was my postdoc. And that uses the idea of words from, spell checking in some sense to denoise the data. And then with a PhD student, we went further in this analogy in some sense, and we realized that we could use topic analysis, which is a method that people use for doing document analysis, where you take a document and you look at the number of words and you look at the frequency of certain words, and then from that you can induce what topics the document is about.
So like if I'm seeing the word car and engine and pistons, I know it's an engine, it's about car mechanics. Whereas if I see different words, and so without actually reading it, I can have a very good idea of the topic.
Exactly, but it's not pure. That is the big advantage of topic analysis on simple clustering is that it could be about cars, and then you could have a whole set of legal words like contract and buyer and so on, and you could realize it was a legal document about cars, and so you would have two topics. And that's exactly what we use now for doing all our microbiome analysis. That is, we have the topics. The equivalent is communities. So you have these like guilds of bacteria which are working together in small communities, and you have the assembly of communities which change. And this has been a huge breakthrough for us, because one of the big difficulties was in bacterial communities, the functions are not always played. The same function is not always played by the same bacteria. So you have synonym. Right. So you have things which are synonymous, and in the same way you have words which are synonymous.
So who's going to do the job today? It could be Al, or it could be Joan.
Exactly.
And in the community, that even among the bacteria, whoever's doing that job might change.
And that plays havoc if you use plain statistical analysis. Because if you're looking at abundances and you want to look at a difference in abundance between two bacteria, if one strain is being replaced by another strain, all of a sudden it disappears. And so statistically, it's a mess. But these topics, if they stay, if you can do a difference between topics, that's a much better unit. I'd say.
So when you were telling me this, I thought you were going to say that the individual bacteria were the topics, but that's not right. What you're saying is that the kind of functioning community is the topic. And that makes sense now, that as long as the community continues to function, the addition or the loss of one or two members doesn't mess it up. It's still the same community. And so it's a much more persistent, I guess, strong and easily detectable, or more easily detectable signal.
Exactly.
Well, that's fair. So, and then, as you said, this turned out to be something very analogous to what is being used in document and text analysis. And was it inspired by that, or was it kind of a parallel discovery?
We started to say, we need to something more flexible than just plain clumps or clusters, and we need mixed membership. And then we remembered, the people in text analysis, and David Bly in particular, they have something called topic analysis. That's really what we need. And then we just could bring to bear that tool and make the parallel. And it's been very, very powerful. That's
a great story. This is the future of everything. I'm Russ Altman. I'm speaking with Dr. Susan Holmes about her work with text analysis, bacterial community analysis. I do want to briefly touch upon your work in immunology, especially since I know a lot of it is quite contemporarily relevant. So what are the challenges in immunology that have caught your attention?
So I'm interested in the immune system because of the dynamics of how, immune system within a person, I would say the immune system is a mini evolutionary system. That is, you have the cells are changing and evolving and becoming different, and you have different types of cells. You have even thousands of different types of immune cells. And the technology, of course, again, we went from very simple technology when you had about 5 or 6 markers and you could use something called flow cytometry, to now we have data which is mass cytometry. And I work with Catherine Blish in the medical school at Stanford on trying to understand the NK cells and how they intervene.
These are very important, just for those who are not immunology experts. NK, or natural killer cells, are a very important cell type in the immune system that are involved in killing infected or even cancerous cells through the immune system.
Right. And so she does a lot of work in vitro, and so we have the sort of cell populations where you do experiments. So this type of experiment, of course, now with COVID, everybody's been listening to other types of immune cells. You've heard a lot about the B cells, so you have T cells. So you have all of these different cell types which exist and create a system. And you can look at various aspects of the system and see how different hosts in different situations, so different subjects, are going to interact differently. So for instance, in pregnancy, women's immune system is modified because, of course, in pregnancy, she has to accept cells which are not herself in some sense.
Right, the
baby. We've done studies of how pregnant women have a different reaction to the flu. And so those types of modifications, natural modifications of the immune system, are very important in also understanding, for instance, women who are pregnant who have HIV, and how the immune system reacts, but how you can reinforce or help the natural killer cells to be more effective.
Yes.
And so those are the types of experiments I'm involved in. And so, of course, these are, as I said, they're not clinical trials. They're very experimental. That is, it's things that happen in the lab, but we're trying to understand the mechanics. And
I'm struck that your description of the immune system started to sound a little bit like your description of microbial communities. And I'm wondering if the ideas of these communities, of these topics that you talked about in the setting of the microbiome, does any of that transfer to the immune system?
Well, the complexity of the interactions does, I would say that. I won't say that it's communities, but I will say that, for instance, I have worked in the cancer environment. We definitely see, you know that some cells, they're what we call the DC cells, cells which are certain type of immune cell which wake up or, facilitate the arrival of other types of immune cells like the T cells. And so there is definitely, I think signaling is a common denominator. And I don't know about communities, but it's definitely about networks and signaling comes in. That is a common denominator. And I would say that the network structure is something that, which we use a lot of. And we have these challenges, of course, because now we're measuring, 70 things, 70 different markers or protein markers on these cells. And we only have a small number of cells, of subjects. Yeah. And so when you're trying to see, does this make a difference? Well, if you only have 10 subjects, but you're measuring 80 things on them, you always will find something significant. And so the challenge is what is meaningfully significant or biologically significant. And that's where the statisticians, we're always seen as the enemy, because every time, my collaborators, show me the data and they say, oh, we have these 3 subjects and they're showing this phenomena. And I just laugh at them and say, 3 subjects, go away, come back when you have 30. And,
right. And that's bad news for them.
Yeah, but immunologists also, originally, I'd say that immunologists work very, very closely and always were trained as doctors. And so everything is done as a case study. So sample size 1.
Right.
And so if they have 3 already, that's, that's 3 times more. So that's already a sample for them. Yes. For us, that's just an anecdote, and you have to go much further. So we've had to push the field.
Well, I'm struck that this reminds me of the very top of our conversation where you were saying you do your analysis, but then you engage the biologists, the doctors, and you get them struggling with the data just as much as you've struggled. And then perhaps some of your perspectives on whether it's a lot of data or a little bit of data is more understandable by them. Yeah.
That's right. And they are doing the analyses themselves very much. And so we go back and forth, and that's very rewarding to empower people. I think that, my side of me wants to teach people how exciting statistics is, whereas everybody thinks it's the black box that they want to do at the end, and they don't want to get involved in it. It's just to open it up and say, it's not that bad.
There you go. Well, with that, I will thank you for your time. I'll thank you, everybody, for listening to The Future of Everything. I'm Russ Altman. If you missed any of this episode, listen anytime on demand with the SiriusXM app.